Hi, hello students. Welcome to all. This is Satyaprasad, Senior Botany Faculty, working in one of the corporate college in Hyderabad. Now I am going to present an important, interesting topic in our NEET syllabus botany, that is the messenger RNA. Before going to understand about the messenger RNA, first of all, we'll have an idea about the RNA. Ribonucleic acid, one type of nucleic acid which plays a very important role directly in protein synthesis, that is the ribonucleic acid. This ribonucleic acid plays a very important role in protein synthesis in different ways, in different forms. Based on this, in the living cells, three types of RNAs are present, appear. That is the mRNA, tRNA, rRNA. Now we are going to understand the details of mRNA. The full form is messenger RNA. Why it is called messenger RNA? Message, RNA which contains the message. What is that message? Genetic message. What for this message? In the synthesis of polypeptide chain, protein. Then this RNA is called as a messenger RNA. So RNA which carries the genetic information from the DNA template is called as a mRNA. Full form is messenger RNA. So where from mRNA is formed? A strand of DNA that is called as a DNA template. So RNA which carries the genetic information from DNA template, it is called as a mRNA. Who first discovered this mRNA? That is the point. Who first reported this mRNA? Jacob and Monod first reported this mRNA. Okay. Then coming to the other features of mRNA. How much amount it is present out of total RNAs? That means around 2 to 5%. Out of total RNAs in the cell, that means mRNA, tRNA, rRNA, out of these three, what about the amount of mRNA present in the cells, living cells? Generally, commonly is 2 to 5%. Coming to its molecular weight, what about the molecular weight of mRNA? Is 5 lakhs Daltons. Molecular weight of mRNA is 5 lakhs Daltons. Out of mRNA, tRNA and rRNA, it is the longest RNA. Among these three RNAs, which one is the longest RNA? Means mRNA. The next character of mRNA is the lifespan. mRNA is formed in the nucleus by a process called transcription. Once it is formed in the nucleus, it is come out of the nucleus and joins with ribosomes and involved in the process of translation, which results the formation of polypeptide chain. Once the polypeptide chain formation is over, immediately mRNA disappearance occurs. Degeneration of mRNA takes place. So the lifespan of mRNA is very, very short. How much it is? It is variable from few seconds to few minutes. The lifespan of mRNA ranges from few seconds to few minutes. And also variable from prokaryotes to eukaryotes also. The lifespan of mRNA in both prokaryotes and eukaryotes is not same, variable from prokaryotes to eukaryotes. Now we will see the lifespan of mRNA in prokaryotes. It is few seconds only. What about eukaryotes? It ranges from few minutes to few hours. Few minutes to few hours. That's about the lifespan of mRNA. Now coming to the few more characters of mRNA. mRNA contains only one polynucleotide chain. It contains one polynucleotide chain. one more important character. When we observe the structure of mRNA, in the structure of mRNA, only one polynucleotide chain is appear. And this polynucleotide chain is straight in appearance, straight in form. It is not folded. It is unfolded. Okay. So when it is straight and unfolded and contains only one polynucleotide chain, no appearance of base pairing. mRNA is a single polynucleotide chain strand. Because of this reason, no appearance of any base pairing. But appearance of base pairing is a rare one in mRNA. It is a specific one. In the process of RNA interference, double-stranded mRNA formation occurs. In the process of RNA interference, formation of double-stranded mRNA takes place. What is the importance in the formation of double-stranded mRNA? It involves in the killing or destruction of the nematode melodygine incognitia. This nematode parasite, it is a root parasite causing a root knot disease in tobacco. It destructs the tobacco plant, reduces the yield of tobacco crops. So RNA interference is a method to avoid, to stop, to prevent the nematode parasite. Okay. In the RNA interference, formation of double-stranded mRNA takes place. When double-stranded mRNA formation occurs, base pairing occurs. When base pairing is there, that is the lethal effect, a harmful effect. It destructs or damages the biological activity. Generally in mRNA, no occurrence of base pairing because it is a single-stranded polynucleotide chain. But specifically, sometimes it forms into a double-stranded exhibit base pairing. If base pairing is there, if base pairing occurs, that leads to the damage or destruction of biological activity. Example, RNA interference in tobacco plant, pest-resistant tobacco plant to damage the nematode parasite, that is the melodygine incognitia. Now, this mRNA's role, what is the role of mRNA? Brings the information from DNA. What for? To produce, to synthesize polypeptide chain. So it contains the information for the synthesis of polypeptide chain. That is why it is called as a information RNA. mRNA contains genetic information for the formation of polypeptide chain. That is why it is called information RNA. And this mRNA is involved in the synthesis of polynucleotide chain. mRNA acts as a template. That is why it is called as a template RNA. Template RNA. That is about another important point. This mRNA is formed from DNA template by a process, important process, that is called transcription. So by process of transcription, mRNA formation occurs. In the transcription, mRNA is formed from DNA template. Okay. That's about the mRNA characters. This mRNA is involved in the synthesis of polypeptide chains. This mRNA is involved in the synthesis of polypeptide chains. How many? One or more than one. Based on this, in the living organisms, two types of mRNAs are there. Monocystronic mRNA and polycystronic mRNA. Based on the number of polypeptide chains produced by an mRNA. mRNA is classified into two types, that is the monocystronic mRNA and polycystronic mRNA. mRNA, which contains the information for the formation of one polypeptide chain, that is called as a monocystronic mRNA. mRNA contains information for the formation of one polypeptide chain. It is the monocystronic mRNA. Where it is present? It is present in the eukaryotes. Monocytonic mRNA appears in the eukaryotes. Other characters of monocytonic mRNA is metabolically stable. It is metabolically stable and can stay for longer time in the body. Okay. Longer time means up to hour only. Okay. It starts protein synthesis. It initiates protein synthesis at one site only. At one place only, this mRNA is concerned in starting the synthesis of polypeptide chain. That is about the monocystonic mRNA. Coming to the another type of mRNA, polycystonic. mRNA is involved, contains information for the formation of more than one polypeptide. It is the polycystonic mRNA. It is found in prokaryotes, primitive. Polycystonic mRNA is found in prokaryotes. It is metabolically unstable compared with the monocystonic mRNA and cannot stay for longer time. That means it is present only for few seconds only. Okay. It initiate the protein synthesis at more than one site in internal position. At any internal position, there is no specific region. At any region, it can start the formation of polypeptide chain. That's about the polycystonic mRNA. So, in this way, based on the number of polypeptide chains formed from mRNA in the living organisms, two types of mRNAs, monocystonic mRNA and a polycystonic mRNA. Then coming to the next one, most important part of mRNA, that is the structure of mRNA. In the structure of mRNA, various parts, various regions appear. Various parts or various regions appear. Now we are going to understand what are the different regions present in the mRNA briefly. Because in our NCERT textbook, they are not given in a specified manner, but they are mentioned in the NCERT book, but not in a particular manner. But it is very important to understand what is the structure of mRNA. When we observe the structure of mRNA, in the structure of mRNA is a straight chain. It has one end called 5' end, another end is called 3' end. Coming to the 5' end of mRNA, okay, this is called as a cap region. This is called cap region. 5' end of mRNA is called a cap region. Okay, what is the importance of this region? This region provides protection to the mRNA from enzyme action. This region is concerned in providing protection to the mRNA from action of different enzymes. That's about the first region of mRNA, that is the cap region. Next to the cap region, the region is called as a untranslated region. The region present adjacent to the cap region is the untranslated region. Untranslated region is also called a non-coding region. Another name of untranslated region, non-coding region. In mRNA, two non-coding regions are present. In the mRNA, two untranslated regions are present. One at the beginning, another one is at the end of mRNA. Okay. So it is present adjacent to the cap region. Okay. What is the importance? It provides support to the initiation codon region. Untranslated region 1 or non-coding region 1 provides support to the initiation codon region, where initiation codon region is present. So this is the initiation codon region. Okay. Why it is called initiation codon region? This region which contains initiation codon. What is the meaning of initiation codon? Codon which is concerned in the starting or beginning of polypeptide chain formation. The codons concerned in the starting of polypeptide chain formation, that is the initiation codon. What is that initiation codon? AUG. Okay. So the region which contains the initiation codon AUG, that is called initiation codon region. What it is role? This region is concerned in the starting of polypeptide chain formation, polypeptide chain synthesis. Now coming to the next region, present adjacent to the initiation codon region. This region is called as a coding strand or a coding region. Why it is called coding strand or a coding region? This region is concerned in the complete formation, total formation of polypeptide chain. Okay, it is concerned in the formation of polypeptide chain. That is about the coding strand or a coding region present adjacent to the initiation codon region. Now coming to the next region present adjacent to the coding region. It is called termination codon region. Why it is called termination codon region? The region which contains termination codons. What is the meaning of termination codons? Codons concerned in the termination of polypeptide chain formation. Codons concerned in the termination of polypeptide chain synthesis, they are called termination codons. What are they? That means UAA, UAG and UGA. UAA, UAG, UGA, termination codons. So whenever these codons are present, formation of polypeptide chain stops or prevented. So concerned about the termination of polypeptide chain synthesis. That's about the termination codon region. What is the next region present adjacent to the termination codon region? Untranslated region 2, also called non-coding region 2. Non-coding region 2. Untranslated region 2 or non-coding region 2. Okay. So we had already mentioned in the beginning, in the mRNA, 2 untranslated regions are present, 2 non-coding regions. Both are present at both ends. See here, first an untranslated region, this one, this is the second untranslated region. Present adjacent to the termination codon region, coding region, termination codon region. What is the role? It provides support to the termination codon region. Untranslated region 2, it is concerned with providing support to the termination codon region. Now coming to the next and last region is the tail region. It is the 3' end. The last region in the mRNA is the tail region. So the 3' end of the mRNA. The 3' end of mRNA is represented as tail region. Okay. It also provides protection to the mRNA from action of different enzymes. Tail region is also concerned in providing protection to the mRNA from enzyme action. Okay, this is the complete total structure of mRNA. Okay, so this is about the complete information on mRNA. Okay, so that's about my presentation. I think you are enjoyed this presentation. Thank you very much to all. Okay, thank you very much everybody.